So, questions from the audience?
I have a question for José about roving in the to adjacent vine. So are you assuming that's root grafting from the infected vine? Does it show that it can root graft?
It could be. That was one of our hypotheses, that root grafting happened on grapes. My understanding and talking also with industry and growers who know way more than me what's going on in the field, it happened, but it's not something very common, mostly with the irrigation system we have, where we have the irrigation really provide in both sides of the trunk. I mean, the roots really try to go deep. This doesn't mean it doesn't happen, but I don't think is the main cause of that's my hypothesis. I don't think root grafting. I don't know, Tommy, you have a hypothesis for that, but
yeah, don't think that's as likely. But when we first looked at this, where most of the new infections were to adjacent vines, it got us thinking about subterranean feeding insects. So we were thinking about phylloxera, different things. But it also fits in with the plant hoppers. So the tree hopper, the nymphs are above ground feeding, as are the adults. But from what I understand, based on the biology, plant hoppers feed, the nymphs are underground. So that would make some sense if they're feeding underground. Roots may not be grafting, but if you have insects down there that are moving around feeding on the roots are quite intertwined and meshed together. And Andrea can talk. So Andrea did, credit to her, the bulk of the work on the sampling, and the plant hopper, you did find a fair number.
We found some nymphs in a lot of things in alfalfa to the vineyards and that kind of thing. And so we were looking at timing. So we collected them throughout the season. And there were nymphs in Delphasidae family. So it wasn't the same ones that Justin had found in Ontario. So it's a different, he had Sixidae, so it's a different family that they're in. We haven't gone to any testing other than doing some collections. We're looking at maybe doing some soil samples to see and putting them in Berlese funnels to see what we can get out of that. But we haven't made a lot of progress yet on any kind of testing with them or anything like that.
So we chose to focus on the tree hoppers, not plant hoppers, because of the evidence, the papers out of the States that alfalfa tree hopper was the important vector. So we weren't focusing on plant hoppers, and there is that issue through the season. They can be underground during the developmental stages. So timing of our sampling should have been adjusted as well. But the idea with the artificial sucrose feeding system, that it's not just sucrose, it's got preservatives and various things in it, but it turned out to be a very good method to screen for the ability. So Asim is back there, my replacement. I shouldn't say that. He's a new person doing whatever he's going to do. But one of the goals is to take plant hoppers and put them in the same system. We didn't include a single plant hopper because we were looking at other groups of insects more closely related to.
And I have to just mention, I didn't have time to provide the information, but when I'm talking about an adjacent plant, yes, adjacent within the row, but also adjacent on the other row. So basically, the plant who was next to it in the other row would be the ones who come infected. So that's why I'm thinking that, and in that case, probably root grafting is not really happening. It could, but I don't think it's really happening. And the other thing that's very interesting is this block was infected, we hypothesized, since 2009. We believe that the material came with replants after the big freeze event that happened in 2009, because you saw the distribution is all over the map. And we didn't really see any spread the 2 or 3 years before we started the rogging. And it was when we were starting to pull out those vines. So to me, it's quite interesting why that happened at that moment. Is that this, we pull out those vines, there's some, movement of the soil, things that you may, that's why I'm thinking it's probably not root grafting.
You had mentioned about the development of the virus getting sufficiently high titers and that sort of thing. I think it's fairly lengthy, actually, and this one as well. So that's another limiting factor.
Justin, Deb, is there anything you want to add to that? Just giving you the opportunity to jump in case. Okay. Yeah.
Jose and Tom, is it possible that virus transfer mechanically, but through the, for example, pruning? So when people do the pruning, then they go to the next vine and then prune that one. So there is the highest chance of transmission. By the time they finish, probably their equipment is clean up to the next. Is that a possibility? Or?
I don't think so. There are really two different viruses, which can be the graft transmissible or insect vector transmitted. And the ones we know on grapes, the graft transmissibility really happens, in the grafting. You have to graft. To the best of my knowledge, I haven't seen any data or anything suggesting that with the pruners, it may happen. We have to think that when we are pruning during winter, even though we are able, to detect the virus in the dormant canes, and it's actually a very efficient way to detect the virus in dormant canes, most of the nutrients goes back, to the roots, and these are, fluent limited. So I don't know the possibility how much you can, transfer with that, but I don't think it has been proven. You can artificially transmit, in experiments, to weeds or things that they have been working on mechanically, but I haven't seen any data on grapes.
And I don't think you're able to do mechanical transmissions from grape to grape where you abrade the leaf or you put carborundum or some other material to roughen it, and you can just apply it. Some viruses you just apply it to the leaf, and you can't do that with these viruses. So that's proof that it's not readily transmitted mechanically, if at all.
I'll just do one question online. I'll come back to you. So 1 or 2 questions online. In your GLR3 replanting study, did you evaluate the effectiveness of the roguing process? According to the literature, proper roguing should include complete removal of the vine's root system. Was this aspect considered or monitored thus for you?
No, in the leaf roll where we were looking into the new plantings, we didn't look into the roguing. That was only looking into stom actually work. Looking the epidemiology and the spread, next to an adjacent block, we didn't do roguing. The standards for roguing coming from work in New Zealand or California, South Africa, they recommend to rogue even 2 adjacent vines in front and behind infected vines, as well as another 2 on the adjacent row. So you are eliminating about 8 vines per vine that you are, roguing. That has come down right now to basically adjacent vines because it's very costly, and they have shown that eliminating just the adjacent vines will work. So, but we are actually having work within this SCAP to do roguing in leaf roll vineyards now.
I was just wondering, what is our experience in BC compared to Nova Scotia? It seems like theirs is mostly coming from nurseries with leaf roll 3. In our situation, is it coming from nurseries, and do we have active vectors? And if so, which are the vectors that are most important in BC?
Would you be able to repeat the question as you answered it?
Can you repeat the question?
Sure. So the question was that it appears that in Nova Scotia, most of the infections with leaf roll virus are arriving from nurseries. How does that compare with BC? And the question is, what is, I guess, the impact of vectors versus nurseries? Do we have both? And yes, we have both. We were able to identify early on sources, some nurseries, the CFIA stopped importations from certain nurseries in France that were too dirty. So they weren't allowed to ship here because there was too much virus infection in them. So there was a lot of initial infection when they stopped restricting leaf roll virus. It was removed in restriction 95 or so, mid-90s. They did a big survey here in the States, and there's a lot of leaf roll virus, so they didn't restrict it anymore. So once the quarantine was off, there was unfortunate quite a bit of rapid spread, not only from nurseries, but then we had flare-ups. If people remember back in the mid-90s, we had flare-ups in mealybug in particular, and they are the more efficient vectors from what we can see. We haven't done the work yet. We'll have to use those little tubes and see. Asim can try the next actual work comparing scale insects and mealybug transmission efficiencies, we call it. We know that the mealybug from past experience is very good vector, and we had tons of them. So we got plants, infected plants from the nursery, so that's bad. That's not a good start. And we had unfortunate flare-ups, huge numbers of, in particular, mealybug. And we had certain areas where it was just flew through. Virus and leaf roll infections just came in like crazy, ended up with some blocks almost entirely infected. So here it's a real mix. We need to look after both. We want clean plant material and we want vector control. As you saw, and Andrea can speak to this, there is a high rate of parasitism. And so, for example, mealybugs, if you stop spraying certain materials, and certainly in organic vineyards, we just don't see mealybug populations the way we do in some commercial, some traditional types of practice. So that's something that has to be considered. There was the one that new records, a lot of what Andrea found, there are new records here, and you found the mealybug parasite.
The mealybug parasitoid is an introduced biocontrol agent that has done quite successful in Canada. And when we were doing the collecting of things, I didn't find a whole lot of them at the time. So I am finding some evidence right now of it being around. I also didn't get the pleasure of being around when we had the big mealybug population explosions. I came just after that. So I don't know if I can say I'm looking forward to maybe finding it one day. Probably you guys don't want me to say that. But in terms of cottony vine scale, that's where most of my parasitism work has come from. And as one of the slides that Jose said, from collections on site and in Oliver, I did get about 80% of the adult cottony vine scale were parasitized. And in one of those cottony vine scales, I reared out 72 individual parasitoids. So parasitoids are little parasitic wasps, if you're not familiar with them. And so they can act as a bank to build up populations. So when they're small nymphs, when the scales are nymphs on the leaves and stuff, they can house maybe 1 to 3, maybe sometimes 6 parasitoids. But as we see the larger adults that have the big cottony egg mass associated with them, they can build up the populations to up to 72 adult parasitic wasps in there. And so if you think about one of these cottony egg masses, like when the scale parasitoids come out of them, how many they can each parasitize. So the control level in there is quite high. And so that was at least 80% I saw in the adult cottony vine scale, and then about 67.5% in the nymphs, which was pretty impressive. I was pretty impressed with that.
Yeah.
97% of the parasitism comes from one species, though. It's there is about 6 or 7 different parasitic wasps that are out there, but the majority of it comes from one certain parasitoid species. So interesting info for sure.
Yeah.
Justin and Deb, from the Eastern point of view, is there anything you want to add to this conversation? No pressure, I'm just.
No, I don't think so. I there probably was a contribution in the very early days of the industry here, in terms of growers kind of propagating from their own blocks, and possibly movement of virus that way, or infected tissue. But I think what brought this to the forefront in Nova Scotia was there was a period of rapid expansion where there was a lot of promotion of the industry to get bigger in a shorter period of time. And so that's, I think, when it really came out on the radar. But we have been incredibly fortunate that our vector pressure is exceptionally low.
I have a question for Justin. Justin, you mentioned your captures of buffalo tree hoppers at the edges of the vineyards. where have you correlate that with, I know in Ontario there is more wild grapes than here. You have quite a lot of population of Riparia or some other wild grapes there. You think that they could be feeding there, or still they are not going to be the main host? Did you look into the wild grape population adjacent to the blocks?
No, we didn't characterize the plant species on the edges too much. My current student is looking into that a little bit more, but anecdotally, when I've been out there sweeping, it seems that goldenrod is a pretty good host, or at least the adults are on the goldenrod when we sweep through it. So that's a good lead. It might, but what we want to do in the future is look at what plants the nymphs are on, and that will give us a better idea of what are good feeding hosts for buffalo treehopper. But I don't think, I mean, we have swept through wild grapes and usually don't catch very many buffalo treehopper.
Hans?
You sort of hypothesized that the nymphs might play a role in moving the red blotch to adjacent plants. So would the nymph have to acquire the red blotch first and transmit it, or has he transmitted from the parent?
Well,
Can you
there's,
repeat the question?
yeah, so Hans is asking if we're talking about transmission of, you're talking red blotch virus by the nymphs of the plant hopper that would be underground. There's two possibilities, and I think it's been brought up earlier that there's possibility of transmission from adults to the nymphs. And there's also possibility, because they're underground feeding, that the grape roots often can get intermeshed. They're not perhaps fusing, but they're close enough that the nymph could feed on roots from an infected vine and then move a bit, feed on one. So it's both are possible. And I think a little more work needs to be done to figure out if we're also getting what they call transovarial transmission. So down the line, that infected female. There are some of the Gemini viruses that are kind of oddballs. They actually are able to infect plants and also the insect vectors. And so it's pretty early on this. Can plant hoppers, can the virus persist in them for quite a long time and also be, I don't know that yet. There hasn't been enough work on this group to figure that out.
The nymph is pretty mobile. Well, underground, stays underground long enough.
So I think we have to refer to Ontario work on that, because the biology is Justin there. Do you want to answer that one, Justin? You've done more on the biology of the nymphs. We weren't looking for them at first.
Well, we have not yet been able to locate the nymphs of the Melanoliaris plant hoppers. There is virtually no literature on what they might look like or what plants they are feeding on. So we're basically starting with taking some clues from a plant hopper in Europe that is similar and can spread I think it's the Bois Noir phytoplasma, if I'm not mistaken.
Yeah.
And in that case, I think that phytoplasma can be acquired by the nymphs on the roots of weedy species, and then it's transmitted from the nymph to the adult, and the adult occasionally visits, a vineyard and feeds on a grape, and then transmits the phytoplasma to the grape. But I was going to say, for GRBV in alfalfa treehopper, it has been shown that the virus is transstadial. So that means the nymph can pass it to the adult, but it is not transovarial. That means the adult female cannot pass it to the egg. But that's in alfalfa treehopper.
Any other questions? Yeah.
that I see they using Movento. Are they spraying it on under vine on the soil, or they spraying it on the foliage?
Deb, with Movento, are you guys spraying it under vine or on the foliage?
My understanding is most growers are spraying it on the foliage. And we're recommending that they monitor quite early in the season. And so when you have, you're at the 7 to 12 leaves, you're targeting then. If you wait, in some cases here where populations can be very high within a block, it is my opinion that if you wait, late enough in the season, then it's I don't think you're going to get your biggest bang for your buck.
Thank you.
We have time for one more question. Yeah.
Why not? Here's the discussion around all the leaf hoppers and nymphs. Has there been any work done then on cover crops we should be aware of that might be also hosts for some of these fires? I know we know that's
The question was, should we be concerned about cover crops or plants in vineyards and how they might play a role in the epidemiology of these viruses? That's hopefully close enough to what you were asking. So yeah, we're always worried about the ground cover species. These ones are quite specific, so it should be looked into, but there's no other known hosts for leaf roll virus, for example, that I'm aware of. And red blotch, it's a little newer, but there was some indication earlier that there was a couple plants that I thought they were worried about in the legume family, I think. So alfalfa and things like that. But I don't know how well that's been worked out. It's unfortunate if red blotch does go into any of those because growers like to utilize legumes for the nitrogen and other reasons. But I think that's still a little bit early to say if that's a big problem or not.